Welcome, everybody. I'm very excited to see all of your faces and your names here at our Hutch at Home series. Thanks for making space today to join us. This has actually turned out to be one of my highlights of each week as I get to connect with you and also with some fabulous scientists and hear some interesting stories about careers as well as some of the work that different people have been doing. And it's nice to see some of you are turning into real Hutch at Home regulars too. So nice to see some of the same folks showing up. This is geared primarily for our teaching community, but we usually have had a few students on as well. So I would like to welcome both the teachers and the students today. And make sure that you keep track of your questions to put in the chat or to ask when the opportunity arises, especially at the end of the session. So today, I'm really excited to introduce the topic of vaccines, technology, and public health. So I'm Jeannie Chowning. I'm the Senior Director of Science Education at Fred Hutch Cancer Research Center. And our speaker today is Juliane Hill, who's a technical applications specialist at Cytek Bioscience. So this is actually a little bit of a different talk for us, but I'm excited to have this special focus on technology and how technology and research kind of work together to advancing our understanding. Okay, so just some quick reminders. Please mute yourself if you're not talking. And sometimes if you come in on a cell phone, it's a little harder to remember to do that. So please do minimize the background noise. Feel free to add your questions into the chat, and then we'll have a feedback form at the end of and then, without further ado, I'd like to introduce Julie Hill. And this picture of you with the umbrella is very apropos for the weather today. But again, we're delighted to have this special talk focusing on flow cytometry. Okay, so welcome, Julie. I'm going to turn this screen over to you.
Okay, thanks, Jeannie. I'm gonna block my video. And oops.
Sometimes you have to click in the slide back with your arrows.
I'm trying. Let's see. It'll go forward. It's not going backwards. Oh, maybe this is where I'm starting. Okay, thanks, Judy.
Okay, we good?
Oh, great. Okay, thanks. Welcome, everybody. Thank you for coming. And I am Julie Hill, as Jeannie said. I am from Seattle. I wanted to start with some of my background. I went to Seattle Prep, and I also went to Seattle University. For any of you who've been around for a while, there's a program called Matteo Ricci, and mine was, I think, the first class that went all the way through. We were 6 years total, 3 years at Seattle Prep and 3 years at Seattle U. Came out with a BA in humanities. And I knew I wanted to go on in science. So then I went to the University of Washington and got a second undergraduate degree in biology and then a master's in toxicology. Here's just a couple hobbies that I have: gardening, knitting. And to be honest, I went for 1 week to surf camp, which was super duper fun. I'm wishing for that kind of escape right now, I think. Then the jobs that I've had are, my technical specialty is flow cytometry. I worked in clinical laboratories. I actually started in a clinical lab when I was in high school. I was 15 years old. My aunt was the supervisor of the cytology department, and I started working for her. I've worked in labs ever since. I've worked at the University of Washington, so in research labs there in academia, also in some biotech labs. when I was at Zymogenetics, I actually, during a couple summers that I was there, I had SCP teachers come and do the summer internship with me. That was very fun. And that was, I think that was maybe where I got introduced to SCP. So what I want to talk to you about is time that I spent at the National Institutes of Health, and then I'll just really briefly talk to you about the technology that I'm working with right now at Cytek. Okay, and so that I don't forget, I put this up at the front. At the NIH, there is a postbac program. I just wanted to plant this seed. I know you guys are high school teachers and maybe some high school students. But the post-baccalaureate program that they have at the NIH is really remarkable. And it's students would spend 2 years there in a research lab, so between graduating from college and then starting either a graduate program or medical school or some kind of postgraduate work. And if anyone is inclined to do that kind of research, the campus is amazing, and it's just really great scientists, of course, to work with. And I highly recommend at least looking into it. If I'd known about it when I was of age, I would definitely have done this. All right, so this is my title. I am going to talk to you about flow cytometry in the context of the work that I did at the NIH. And so I'm going to start by telling you a little bit about the NIH because I didn't know anything about it when I went there. So there are 27 institutes and centers. I worked in the National Institute of Allergy and Infectious Diseases. Tony Fauci, my hero, is our director. And there are two parts to it. There's the extramural part of the NIAID, and that's the granting institute. So they give you grants if you're writing grants. And then the intramural part, which is where I worked, that is internal at the NIH, and this is internal research on campus. So I worked in the Vaccine Research Center. And the Vaccine Research Center, VRC, as we fondly refer to it, it was built in the year 2000. And I'm just going to read you the mission. It was to conduct research that facilitates the development of effective vaccines for human disease. The primary focus of research will be the development of vaccines for AIDS. So that's that was the goal of the center, but all kinds of infectious vaccine research was going on there. And so the VRC is directed by John Mascola. There are different laboratories and programs within the VRC. And then I was in the immunology laboratory. Within the immunology laboratory, I was in the immunotechnology section, and that was led by Mario Roederer. So I was actually invited by Mario to come and work in his flow cytometry lab. And the goal, my project, flow cytometry, for those who don't know, it's a fluorescence-based technology for characterizing cells. Now, my project there was to develop a 5-laser 48-color cytometer. When I arrived at the VRC in 2013, we had systems with 3 lasers and 18 colors. And by the way, there are 7 flow cytometers within the Vaccine Research Center, which lets you know the priority that they put on flow cytometry as part of the one of the research tools for vaccine development. Okay, a year after I got there, we had 5 laser systems and were able to measure 28 colors. And then for the rest of the time that I was there, our hardware was more capable, but still we were only measuring 28 colors. Now, what in the world does all that mean? So what is flow cytometry? Well, it's a really cool technology. We use it to answer biological questions using physics and chemistry, engineering, and math. So there are lasers, there are light detector devices, there's fluidics, there are fluorescent dyes, there's hardware, there's software, there's data analysis. So in my opinion, I'm kind of biased, but I think this is really kind of a perfect STEM technology. So what exactly is it? So the flow of flow cytometry has to do with a fluid stream. So a fluid stream carries cells through the cytometer. And then cells are what are measured and identified. And the thing that's measured is photons of light. Okay, so What do we do with cytometry? We identify, characterize different cell types. We can count cell populations. And what do we want to do this for? Of course, to study biology. So it's used really extensively to study the immune system and associated diseases, autoimmunity, cancer, viral infection. And we can measure, identify, characterize all different cell types. So different cells express different antigens. So this is a protein that's on the surface of the cell. And then we use antibodies that are specific to the antigen to identify the antigen. And each of the antibodies then has a fluorescent tag attached to it. And this fluorescence is the light that we measure on the cytometer. Okay, so antigens, these are structures on or in a cell. They're commonly proteins. And again, different cells express different antigens. So the antigens expressed by a cell identify that cell and also tell you something about it. Is it active? What's it doing? The naming of antigens is kind of curious, but we name them with what we call CD numbers, and CD stands for cluster differentiation. That's kind of a whole story in itself about how CD numbers came about. So I'll leave that for now. Then antibodies they have on them specific binding sites. And these binding sites are very specific for binding to particular antigens. And the binding between antigens and antibodies, it's a lock and key kind of fit. Oh, and by the way, Jeannie, I'm not monitoring the chat, so feel free to interrupt me anytime with anything that comes into the chat. And
sure, I'm watching it, so I'll let you know if there are questions that come up that seem timely to what you're discussing.
Okay, sounds good. And I guess I'm willing to try if people want to just unmute themselves and interrupt and ask questions. We can give that a try. If it turns out not to work so well, then we'll go back to just chat, but feel free to use either mode. Okay, so then the reagents that we use in flow cytometry are antibodies that are, we say, conjugated to a fluorescent dye. So here is a cartoon of an anti-CD8 antibody with its purple fluorescent tag. And here's a cartoon of an anti-CD4 antibody with its orange fluorescent tag. And so here's our cell. Here is the CD4 antigen on the cell. So CD4 is actually an antigen for T cells, and it's specific for helper T cells. And I know a number of you guys have heard about these, and we in other Hutch at Home seminars, these have been discussed. So your antibody dye reagent binds very specifically there's one that binds very specifically to this CD4 antigen. And then we measure the fluorescent light, the fluorescent tag that comes off of the antibody that's attached to this cell when that fluorescent tag gets excited. And it's excited by a laser beam, and we'll talk about that in a minute. Okay, so what we do then in the lab is we mix cells with antibody dye reagents, and we call this staining. So the cells are stained with antibodies that have fluorescent tags on them. And again, different cell types express different antigens, and then antibodies are specific for those antigens. The cells then are counted one at a time in the cytometer, and the photons that come off of the fluorescent antigen or antibodies are measured, and then those tell us something about the cell. Now, I've pictured here, all of my cartoons show just one antibody binding to one antigen on the cell. In reality, the cell is decorated with antigens and can also be decorated with antibodies. So there's almost always more than one antibody bound to the cell. So usually more than one color is associated with cells. So let's break down here what happens at the measurement step. Okay, we've got, here is our cell that's been incubated with fluorescent antibodies, and we're going to send it past a laser beam, which excites the fluorescence, and then that light is measured by a detector. And the detector, light collection detectors convert photons to electrons. And then those electrons are digitized, and that's what becomes our data. So this is a cartoon of a typical flow cytometry data plot. Now, so fluorescence is what's being measured on each of the axes. So in this case, orange fluorescence. So some of the cells that went past that laser beam were negative, had no fluorescence on them. Each of the circles here represents a cell. And some of the cells had a lot of orange fluorescence on them. Some cells had purple fluorescence on them. These two cell types are different from each other. If there were cells that had some of both of those antigens on their surface, they would come up over here, they would be double positive. Also, if there were some cells that had, so these cells say had, I don't know, let's say 50 antigens on their surface, and that gave them this much fluorescence. If there were cells that had only 20 or 25 antigens on their surface, they would have slightly lower fluorescence. And all of these different groups, then we call populations, and they all are meaningful in some biological way. Okay, now we're going to break down what happens here at the flow cell. Okay, here's our test tube with cells. These are going to be blood cells. And here I've enlarged a couple of them. This one is a CD4 helper T cell, and it's got its antibody with a fluorescent tag on the surface. And again, I've only drawn one, but this really is decorated with a lot of CD4 antigens on its surface. And then here's our CD8 cytotoxic T cell. It's got its antibody with its fluorescent tag. Again, there's more than one on the surface here. And then these cells, they're in a saline kind of solution. They're going to go into this chamber called a flow cell. And so the cells will go into the flow cell, and there's also, here's our laser light source, and this is going to excite the fluorescence that's on the antibodies that's on the cells, that are on the cells. Okay, so in this flow cell, in this chamber, there's something called a sheath fluid, and that surrounds your sample that's going into the flow cell. The sheath fluid, it's usually saline, it could be distilled water, it's some kind of a liquid like that. Often it's saline. And what happens is your cells in their liquid stream are going into the flow cell. The sheath fluid that surrounds those cells squeezes the cells so that they move into, they get into line. So they move to one at a time. They go past the laser beam. So the sheath fluid does what we call hydrodynamically focuses. So hydrodynamic focusing, it's a physics principle, and that takes place in the flow cell, squeezing the cells, your cell sample into a line. So cells go one at a time past the laser beam. So each cell is individually interrogated. We call this the interrogation point. There's a play about that. At the interrogation point, then, fluorescence on the cell is excited by the laser beam, and then that fluorescence goes off and gets collected at the light detectors. One of the other measurements that can be made here is called light scatter, and there's two types of light scatter. One is forward light scatter, and that is a measurement relative to cell size. And then there's another measurement called side scatter. And that is a measurement that's relative to the internal complexity of cells. So then here's what data looks like that comes off the cytometer. And this is this is a scatter plot. So the x-axis is forward light scatter. So this is measuring, again, a relative measure of cell size. And on the y-axis is side scatter. So this is a relative measure of internal complexity of the cells. And so this down in the corner is just debris. But here, this population is lymphocytes. And this population here is monocytes, and this population here is granulocytes. So this is human whole blood, and this is what it looks like by scatter on a cytometer. And you can see the populations all really separate out quite well. Lymphocytes, which are small and mainly just a nucleus, have a smaller forward scatter, smaller size. Monocytes are a little bit larger with a little more going on in the cytoplasm, and you can tell that by their higher side scatter. And then granulocytes have a lot of organelles and larger kind of globular nuclei. So granulocytes are larger and have higher side scatter. If any of you were at Nina Salama's talk a couple weeks ago, she spoke about sorting her Helicobacter based on side scatter. And by side scatter alone, she was able to see differences in she had Helicobacter that were straight and others that were curly or curved. And just by side scatter, she was able to tell those two populations apart on a cytometer. My cat just woke up. He might contribute to the talk. Okay, so then here is the fluorescence measurements that we make from a cytometer. So this is our CD4 antibody measuring, or our antibody measuring the CD4 antigen on the cell. So these are our helper T cells. And what's measured is the fluorescence. So this axis is, again, measuring fluorescence. But this group here these are our CD4 positive helper T cells. Every dot is a cell. One of the other things about flow cytometry is that you can collect a lot of data quickly. So I would say typically samples will run through the flow cytometer at about up to 10,000 events per second. Probably 5 to 10,000 events per second is really common speed for collecting data. And so think about that. Each individual cell each cell is being individually interrogated at the laser at a rate of 5 to 10,000 events per second. So it's a pretty fast processing of that data. Okay, then here's our CD8 cytotoxic T cells, and their fluorescence is measured here. So we have some that are brightly fluorescing, and then some that have fewer receptors or fewer antigens on their cell surface.
Julie, there was a question in the chat about what makes up the debris.
The debris. So it would be like a lot of cell bits. This came from whole blood, so some of that could be red blood cells, some of it could be platelets, but I would say mostly it's cell stuff. Just parts of cells. Mm-hmm. Okay, the other thing that, so the flow cytometer we were just talking about is called an analyzer. There's another type of cytometer that's called a cell sorter. And so that does all the exact same analyzing, but then also you can sort cells. And the way that happens is that the cells here they are coming one at a time past the laser beam. Actually, this looks like about 6 at a time, but really it's one. And the stream that they're coming in is vibrated at a really high frequency, so that at a particular point, it breaks off into droplets. And the system is arranged so that when a droplet breaks off, there's a single cell in that droplet. Then beyond the droplets are 2 charged plates. And the cell then, right before it's going to break off inside its droplet, the stream actually is charged, and the stream will be charged based on where that cell has been designated to go. So you collect cells in different, you can collect them in test tubes, you can collect them in plates, you can collect them all in different ways, but you charge the cell depending on where that cell is supposed to be collected. So you can get very specific cell populations collected by sorting them, and then take them to the lab and do other research. So that's an important part.
We
Yeah.
had a question about the side scatter. How come larger cells have higher side scatter?
Because so most of them have a lot more going on inside the cytoplasm. So there's more things in there, mitochondria, lysosomes, organelles, just a lot more
To bounce off
stuff.
of. Yeah.
Yeah, exactly. Right. The light bounces off of all the things that are inside there. I have a friend of mine has a demonstration, a side scatter demonstration, and she uses a Christmas ball. She uses a Christmas ball that's just plain, there's nothing on it, and shines a light on it. And then she gets a Christmas ball that has a whole bunch of like stuff on it and shines a light on that. And the light scatters all over the place. So that's just a visual of that. But yeah, there's just lots more inside the cell for light to shine off of, scatter off of.
Thank you.
Mm-hmm. Okay, so now I've given you a little bit of the fundamentals of flow cytometry. So now I want to bring us back to the vaccine development and just talk about some of the work that I did at the NIH and how cytometry and vaccine development, especially HIV vaccine development, kind of co-evolved. So just to remind people, there were two good talks here during this seminar series on HIV, and I'm going to just hopefully complement rather than be redundant about some of the things that were said. But one of the things we know about HIV is that it's been really hard to develop a vaccine. And so some of the reasons for that are because our natural immune system is not adequate to clear the virus. Also, and this is the huge part about it, the virus infects and destroys the very cells that mount an immune system or an immune response to viral infection. So these CD4 helper T lymphocytes are the ones that the virus specifically attacks. And we've just seen that you can very readily measure these cells using flow cytometry. Then another thing is that the virus evolves very quickly. It mutates a lot, and so this confounds treatment efforts. Now a little more about HIV. So it's a retrovirus, and then this is just a cartoon of how it infects a cell. So here's one of the viruses, and it binds to the cell, and it actually binds specifically to the CD4 antigen on the cell. We'll talk more about that in a minute. And then the viral envelope merges with the cell envelope, the host cell envelope, the membrane. And then the virus is injected into a cell, its coat comes off, and then its contents spill out into the cell, the cytosol of the cell. So the components inside this, inside the virus are, or inside the envelope are viral DNA and a reverse transcriptase and an integrase. And so these things allow the viral RNA to be reverse transcribed into a proviral DNA. The integrase then helps that proviral DNA be integrated into the host DNA. And then when the host DNA is replicated, the virus viral RNA is transcribed and uses all the machinery of the cell to reassemble itself, and then out it goes to infect another cell. So we're going to talk a little bit about, we're going to take a close-up of this part right here, where the virus binds to the cell. So it was discovered that not only that the virus binds to the CD4 antigen on the cell, but it also needs to bind to another antigen, what we call a co-receptor, that's somewhere near CD4. And the binding of these two things is necessary for infection. Oh, and by the way, I'm not an immunologist. And I don't study HIV. My technical specialty is flow cytometry. So just so you know. But I've done worked in a lot of immunology labs because flow cytometry is so critical to that work. Okay, so the two co-receptors, one is called CCR5, this is on T cells. One is called CXCR4, this is on monocytes or macrophages. So, and both of those two types of cells, T cells and monocytes, have a CD4 antigen on them. So the virus can attack both of those cells. So here then is this part right here is called the viral spike, but here's the viral envelope. And then the virus is made up of, the spike is made up of a couple proteins, this glycoprotein 41 and glycoprotein 120. And then part of the GP120 is this piece called a variable loop. All right, here's your cell membrane here, your poor unsuspecting cell that's going to get infected. So here is our CD4 antigen. The virus, the GP120 binds to CD4. And then this variable loop part of the virus has to bind to the co-receptor. So either CCR5 or CXCR4. So once these two bindings have taken place, then the spike changes its conformation and starts the process of merging the viral membrane with the host cell membrane. And just the point here is that we can measure the CD4 antigen and the co-receptor antigen on cells, so we can very readily find these cells that the HIV wants to infect. All right, so now I'm going to just go into a little bit of the history of flow cytometry technology development and kind of as it followed along with the HIV epidemic. So in the early 80s, flow cytometers had one laser and 2 to 4 colors you were able to measure on these systems. In the clinical labs, because we knew that CD4 was the target of the HIV, of the virus, there was a clinical test that was very important to monitor patient health, and that's called the CD4-CD8 ratio. And so what that test is we would incubate cells with CD4, antibody to CD4, an antibody against CD8, and then measure how many cells we got in the result. So in a healthy human, you typically have twice as many CD4 positive T cells as CD8 positive T cells. So a healthy ratio of CD4 to CD8 is 2 to 1. But in patients with HIV who are infected with HIV, the virus would attack and kill their CD4 cells. So over time, that ratio would be inverted. So you'd have less and less CD4 cells, more and more CD8. So you would get inverted ratios. I was in a clinical lab at the time, and we CD4-CD8 ratio measuring was a lot of the testing that we did in our flow cytometry lab. The other thing that we would do is measure the absolute count of CD4 cells in the blood. And once that count got below a certain number, then patients were eligible for antiretroviral therapy. So these measurements were critical to patient monitoring and to treatment. Okay, so then flow cytometry technology development over time. In the early 90s, some very clever scientists realized that all the measurements we'd made before this were on the surface of the cell. So we were just binding with antibodies, we were binding surface antigens. And someone went, hey, if I could carefully poke holes in the membrane of the cell, we could get inside and we could measure proteins that are inside the cell. And so we started measuring cytokines. And what you might know about cytokines, first, they're a chemical for signaling between cells. They're kind of the language of cells, how cells talk to each other. The thing is, cells secrete cytokines, so they don't stay inside the cell. And if we wanted to measure those, we had to figure out how to stop them in place. And so that was figured out. And people use a reagent. It's called a Golgi plug or a Golgi stop, and it keeps the cytokines inside the cell around the Golgi, and then we can get antibodies into the cell through holes in the membrane, and the antibodies then can bind to cytokines, and we can measure those. So when we started measuring intracellular cytokines, then that let us know something about the function of cells, or were they actually functional? So could they make cytokines? Were they making less cytokines or more cytokines based on an infection state? Okay, so then as laser and dye technology advanced, then we had more lasers, we had more fluorescent colors. We could define way more white cell and other cell type, other cell subsets. Flow cytometry is one of the main diagnostic tests for lymphomas and leukemias. So flow cytometry helps to diagnose those diseases. So then, along with measuring surface proteins and cytokines, we started also measuring transcription factors. So someone thought, okay, if we can get inside the cytoplasm of the cell, maybe we can also get inside the nucleus and measure transcription factors. And so that is something that we were able to do then too. And chemokines, another type of cytokine, chemokine receptors. So we could start to get more and more information about the cell and what it's doing and who it is and where it's going. And all this has helped to define and characterize cells as different types of treatments are being invented and tested and such. So then we've got 4 lasers and 18 colors. And then someone came up with a really clever way of working with the software so we could do what's called index sorting. Now, all along we could sort cells, but you would get mainly a big population of cells. Or once you sorted it, you wouldn't really know what you had. So you would sort it based on characteristics by flow cytometry, but then you didn't know for sure what you had in your tube afterwards. Now you can sort into 96-well plates, single cell sort, and then you can use those cells for gene profiling, for cloning. Sorting can happen at 20,000 events per second, 20,000 cells per second. So again, at that speed, you put a single cell in each one of the wells of a 96-well plate. I think some sorters can even do 384-well plates. And so what index sorting means is that you can have the fluorescent profile of every single cell that's in a well. So before index sorting, you would sort into the wells and you would say, okay, well, this is what I think I was looking at in terms of the characteristics or the antigens that are on this cell, but I don't really know what it is. So with index cell sorting, you could identify a particular cell in a plot, in a cytometry plot, and it would tell you exactly which cell that was in your well. So this is an example from a colleague at the NIH. And this example and another example I'm going to show you are using non-human primate peripheral blood. And both of these experiments used cells from rhesus macaques. I'm not going to talk about animals in research, except to say it's important, and there's a lot of oversight, and studies are very controlled, and there's a lot of discussion, and you have to write protocols, and it's an important part of research, and also respected, and not sure what more I want to say about this, but it's an important part of research, and also very carefully done. Okay, so in this case, this friend of mine, she is studying B cells, And she is trying to identify specific antigens or specific antibodies that B cells make. And she is studying, so she's working with animals, with rhesus macaques that have been infected with SIV, so the simian immunodeficiency virus. They're making particular antibodies, the B cells are. So she is sorting B cells, sorting very specific B cells, and she's trying to find the ones whose antibodies will bind very specifically to the CD4 binding site on the SIV virus. So the spot where that virus is going to bind to a cell, she's trying to find that specific place by sorting antibodies that are, or sorting B cells that are making antibodies to that very specific region on the virus. So what she did with she stained her cells, and she's had CD4 and CD8. The numbers here, these are the names of the fluorescent tags that are bound to the antibody. So don't worry about that, but those are fluorescent names basically. So she wanted her cells that were not CD4 or CD8 positive. So these end up to be, so these are lymphocytes, and they're not helper T cells, they're not cytotoxic T cells. And she's trying to find, remember, B cells. So then her next staining is CD3, which is an antibody against T cells, all T cells. So she does not want T cells. CD20 is an antibody for B cells. So that's what she wants. So she grabs these B cells, and then she looks at a very specific maturation of the B cells. So IgG and IgM are identify types of B cells. So she wants IgG positive B cells. And then from these, she comes over and looks at, she has probes that are, her probes are again very specific. They mimic the part of the virus that she wants, or the HIV virus or SIV virus that she wants to bind. And so she has a competitor probe so she can get rid of things that are not specific. And then she has a probe that is specific for this CD4 binding site on the virus. And then that is conjugated to a fluorescent tag. And so then this very small group of cells are the ones that she is sorting into 1 cell per well into 96 well plates. And then she takes these cells and she's able to express the antibodies, and then she can make more of the antibodies. And then she goes on to do other specific binding assays to see if they really bind specifically and tightly to the virus, where she hopes they do. And this is a way then that she is contributing to vaccine research. If she finds an antibody that binds tightly, they test it out as a vaccine. Okay, so that's index cell sorting and how it's used in HIV research, SIV research, ultimately HIV research. So now here we are to my project, and we've got 5 lasers, and we can measure 28 colors. And then in 2017, Cytek, a company, came up with a cytometer called the spectral cytometer. So now this is the latest technology in cytometry right now. With a system that had 3 lasers, they were able to measure 25 colors, and this was unprecedented at the time. Really, you could not get up to 25 colors unless you had 5 lasers. So then last year, the company came out with a 5 laser system, and they've measured 40 colors. So they're really advancing the technology even more. And I think I'm going to skip this part, but the point of spectral cytometry is that you measure the entire fluorescence emission of each of the fluorophores rather than just grab a small portion of that emission. So that is spectral cytometry. That would be probably the topic of its own seminar. And then back to HIV. So here is another example. This again is non-human primate peripheral blood. And this experiment was more than 11 colors. And so first, the cells that are CD4, the CD4 positive helper T cells, and also cells that are positive for CCR5, so the co-receptor. Cells are gated for these two antigens, or they're identified to have these two antigens. And then here, this animal, at a time when this animal was uninfected, here we're looking at other antigens. It doesn't really matter what the names mean, but these two antigens together identify this group of T cells that are memory T cells. And if people attended the Immunology 101 seminar that Alicia gave, she spoke briefly of memory T cells and how these are ones that help B cells make antigens. So these are important cells for the immune system. And in this animal that's uninfected, this healthy animal, what you can see is there are these different populations of T cells, different groups of T cells, memory T cells. I'm just gonna call out this group here, and because these are very important, these are the exact ones that the HIV or SIV virus attack. So then sometime later, after this animal had been infected with SIV, the blood was drawn, the same test was done, and what you can see is that these cells are completely gone. And this is really, I know this is kind of a busy slide, but just the point is that here are the cells that the virus attacks, and it's very clear. And if so if then we have a good vaccine against HIV, that you would use flow cytometry to look at the cells before the vaccine is given, look at the cells after the vaccine is given, and then over time, and see what happens with these cells. Are they always here? Do they go away and come back? So if over time after a vaccine was given, this was the result you saw, you would say, I know this vaccine is not effective. If the cells stay and they don't go away, or even if they go away and come back, if they come back especially, you would say, okay, looks like this vaccine is effective. So this is by identifying the cells, characterizing the cells, looking for them over time. This is how flow cytometry is used in vaccine research. And then here, just to give a very current example, flow cytometry is being heavily used, of course, to study SARS-CoV-2. This is a study by Andrea Cosarizza in Modena, Italy, the place of the first COVID-19 infections in Italy. And so they made observations of blood from some COVID-19 patients, and what they saw, they looked again specifically at helper T cells, CD4 helper T cells. They saw that CTR means control. In infected patients, the number of these cells was decreased, and same with CD8 T cells. And then the other thing that they noticed about these cells, or that they measured and found about these cells that remain, is that their cytokine production was greatly increased. And so, it correlates with the cytokine storm that we hear about that patients are suffering from with this infection. So another example of flow cytometry being used in immunology, in vaccine research, all the time, every day. And so just in conclusion, flow cytometry, I think it's an ideal STEM model, and it has substantially informed our understanding of cellular responses for vaccine work, immunotherapies and cancer. It helps us diagnose blood cancers, and in general, it's used for understanding and advancing immunology and biology. These are my colleagues from the Vaccine Research Center. And I'll just leave you with that. So thank you.
Hey, thank you so much, Julie. This is very informative, and we are ready to take your questions either via chat or unmuting yourself. So please go ahead and feel free to jump in.
And I'd be happy to hear if that was totally overwhelming. I would love to know where to expand on things, because I would like to make this talk user-friendly.
Can I ask a question?
Of course.
Okay. We met a long time ago, Julie. I'm Sandy Porter with Digital World Biology and BioLink, now Innovate Bio, the community college biotech people. And I was wondering if there are examples of flow cytometry data that you can possibly share that biotech students could use, and analyze from home. We could share it on our website and it wouldn't have to be identified or anything, of course.
Cool. Yes, absolutely. In the work that I do now, so as a technical application specialist for Cytek, when people buy our spectral cytometer, I go and train them. If people are interested in the cytometer, I give them demos. But one of the things, we have what's called a dry kit. And so these are cells that have been stained and fixed and lyophilized. And we have a 3-color experiment, we have a 16-color experiment, we have an 18-color experiment. We run these at every training and at a lot of demonstrations. And so those are just, experiments that easily I could pull a couple of those together and let you use the data from those.
That'd be wonderful. And I think we're connected on LinkedIn still, or I can get your contact information, Jeannie.
Yep. Yes. I think that would be useful for actually a number of different features, so we can make that broadly available. Julie, there's a question in the chat that said, did you say earlier that you were tasked with creating a system to do 48 colors? Have you accomplished that, or was that the 40 color one you mentioned?
No, thank you. Yeah, thanks for that question. So we have, the system that I was working on has not reached 48 colors yet. And the system I work on now, it's a different cytometer using a different type of detection technology. So no, we did not reach 48 colors. They still have not made it to 48 colors. I think they're still just at 28. But with this different technology, it's easier to get more colors. And so I'm pretty sure that their 50 colors will come out at some point. I don't know, within, I'd say, a year or two. But from not the cytometer I worked on, but from the newer technology.
Julie, I have a question. This is Jan.
Hi, Jan.
Hi. Can you give an example of what kind of experiment would require 50 colors? Would it be like a cell assay with multiple cell types doing multiple things? What do researchers typically use, like a 30 color or a 40 color right now? What kind of assays do they do with that many colors?
Good. So the 40 color experiment that we ran in my company, it's a general immune profiling assay. So it's T cells and monocytes and B cells, so lymphocytes, monocytes. That one's run on PBMC, so peripheral blood mononuclear cells. So it's lymphocytes and monocytes, and then intracellular cytokines and some transcription factors. So it's looking at kind of in depth at the just general immune profiling. A colleague of mine from the NIH, he has 3 28 color assays. One of them is specifically looking at T cells, identifying T cells and all different T cell subsets. One is a functional, he's got T cells and cytokines, and actually the T cells is T and NK cells. And then he's got one that's cytokines, so kind of T cell function. And then he has another one that's monocytes and dendritic cells. And so a lot of them are just general immune profiling. I would say that's the main use of the larger panels.
So basically just being able to identify a lot of different cell types and subtypes, and then things like cytokines and transcription factors. How do the controls work on that? It always seemed to me like with that many colors, if you have to have a single color control for everything, you know, your number of controls could be bigger than your number of experimental samples. Is there like an easier way to do the controls now?
So the controls are, you know, of course, the key to your experiment. So you still have to have a single color control for every color that's in your experiment. You can do things like if you do your experiment one time, it's really good, all your controls are great, you can store them. And you can store them as reference controls, and then you can use them more than once. So you could run all the controls, they're really good, you save them, and then next week you could come back and just have an unstained and a full stain sample and use all those same controls and run your experiment again.
Cool, thanks.
I actually put in, you had shared with me some background in flow cytometry from the Thermo Fisher site. So I pasted that into the chat for people if they were interested in learning a little bit more. We're almost out of time here, and I do need to tell you about what's coming up next. But just really quickly, because I know there's a lot of biotech teachers on the call, could you talk a little bit about how you've been involved with biotech education and partnerships with teachers, and what's possible there?
Yeah, thank you. So I don't know if Tammy Caraballo is on.
I don't think she was able to join us. So
okay, that's too bad. So Tammy, I can't even remember how I got hooked up with Tammy, but she teaches high school at Glacier Peak, although this is her last year. She was fascinated by flow cytometry, and like I said, I don't remember exactly how. If she heard a talk or if she came and worked in my lab. I think she might have been in the lab. And I worked with a group, Ger Vandenang, and he had a cytometry company in Lake City. And we put together a workshop for teachers, high school teachers, I think it was high school and middle school teachers and a couple students. This was a week-long workshop, and we built cytometers. And so Tammy was part of that, and she actually just loved cytometry and kept up with that. I have always, when I've been in labs, I've let teachers come and students come and run experiments or just learn about the technology. So,
do you think Cytek will be open to having some teachers visit, perhaps?
So Cytek is based in Fremont, California.
Oh, okay.
The thing is, and so I don't have a lab here. I work from home when I'm in Seattle, but easily some of the places where we have a system, and the place I'm thinking of, although I have not asked them at all, though, is Benaroya Research Center. They might be open to having teachers come and do some kind of, let me do some kind of work with them there.
Thank you. Would you mind stopping your screen share so I can share just a few slides at the end? No worries, no worries. So while I'm doing that, please everybody give some appreciation to Julie, either in the chat or with some emojis, or you can unmute yourself for a second to give your appreciation, and I'll get this last slide up here for you. All right, thank you so much, Julie. A couple of you had asked about recordings and schedule. This is on our SEP News and Events page. So the upcoming ones are on top. A little lower down on the page are the recordings. And I put the link also into the chat earlier on. So everything so far is posted there, but I don't know how much longer my storage space will last. So just trying to keep everything up as long as I can. And that'll include this session here. Next week, we have Rob Bradley. He's a professor at Fred Hutch, faculty in public health and basic sciences. And he'll be talking about kind of the work that he's been doing with RNA. And a lot of what we teach about and learn about in high school is the central dogma of DNA makes RNA makes protein, but RNA actually ends up being a molecule that can be used for a lot of other things and important in a lot of other ways. And so his talk will address some of that. So I think you'll find that really interesting. So I hope you can join us for that. And then lastly, feedback form. This link was also pasted into the chat. If you want to scan, this is also a link to our feedback form. I appreciate everybody coming here, and hope to see you all next week. And thanks again, Julie. That was wonderful. Thanks, everybody.